get started. My name is Dr. Kelly Kingen. I am an assistant professor at UB North in Small Island Sustainability. And it is my pleasure this morning to introduce today's distinguished speaker, Dr. Theodora Pinou. She is a professor and chair of biology at Western Connecticut State University in the United States. Dr. Pinou is an accomplished vertebrate zoologist and educator whose academic career spans over 3 decades. She earned her PhD in ecology and evolution from New York University, where she also completed her master's and bachelor's degrees. Her research focuses on turtle, snake, and fish ecology and mechanisms for enhancing STEM education. She serves as facility curator of the H.G. Dowling Herpetological, which is reptiles and amphibians, collection, and is a curatorial affiliate at the Yale Peabody Museum of Natural History, where she worked after obtaining her PhD. Her scholarly achievements include peer-reviewed publications, multiple book chapters, and contributions to conservation science through extensive grant-funded projects. Her research has taken her from the Caribbean to the Mediterranean, exploring animal symbioses, invasive species, and the environmental impacts of human activity. Please join me in warmly welcoming Dr. Theodora Pinou, who's visiting us while on sabbatical to help build capacity in the Bahamas by sharing her talk on applying CURE, which is course-based undergraduate research experiences in creative activity and scholarly research. Welcome.
Great. Okay. So Kelly, you're going to help me here so that I can see, how do I change?
Oh, I got to get back to the other screen. Just a second, guys.
And thank you so much for that warm introduction. Boy, I was like wondering, who is that great person? So thank you very much. And thank you very much to the University of the Bahamas for welcoming me today. And I'm looking forward to getting to know the campus, getting to know the faculty that are here and the administrators and students, and look forward to, collaborating some more with everyone here. Okay, so today, the purpose is not so much to talk about my research, but really to talk about teaching and faculty and strengthening the scholarly enterprise, right, for faculty. So I'm going to talk a little bit about some best practices in teaching and learning, and hopefully by the end of this talk, you'll see how university faculty at smaller schools can increase their scholarly output and also engage their students better or more. All right, so I'm going to start with this slide here. Oh. A moment. It just disappeared. Oh. Thank you. Gotta love technology.
I think we might have moved a cord or something. Let's see. Let's try this again. Extend it. Just a second. Sorry. Change off of extend. Okay.
Yeah, you're still muted.
Okay.
Okay, great. All right. So, gotta love technology. But anyway, we were looking at this slide here, and I was just posing the question, does this look familiar, right? Students that are kind of disconnected from the classroom, and examples where you're speaking as a faculty member and you turn around and everybody's totally asleep or someplace else, right? So, right. We all have, I think this is universal amongst, instructors, teachers, professors, this, what I call the student engagement challenge, right? Where students are coming into your classroom and you evaluating them are feeling like they have limited foundational skills. Their coursework is, not up to par. They don't participate in the classroom. There might be a lack in their depth of understanding of the material, and the changing technology and the advancement of AI is creating this gap, right? With the students who can do it and the students who can't do it. So there's this fast moving technology landscape. So all of this is sort of lending itself to what I call the student engagement challenge. And so, this leads to this, right? Does this sound familiar? Do you feel this way as a faculty member where you feel you're exhausted? You're dealing with the students. You feel like there's lots for you to do. You can't keep up. You need to be cloned. And it just creates this real challenge on both ends. The students are disenfranchised, or the students are not engaged. They're coming because they feel they need to be there for whatever reason. And you're struggling because you have all of these responsibilities and you're not connecting with your students. So what can we do about it? I call it the faculty burnout challenge, right? I sort of bend what I perceive as responsibilities of faculty in four categories, right? As teachers, right, where we're evaluating our students, as mentors, where we're nurturing them, as researchers, because, right, we're expected to do research. And lastly, or some creative activity, and lastly, service, which is another component of what we do. And that's four categories, and it can be daunting. Wouldn't it be nice if we could figure out a way where we could combine three, and this way from four responsibilities, we're down to two responsibilities, right? So my goal today is to sort of share some ideas of how that can be done. All right, so what do I plan to do? Cover some pedagogical theory, right? Some best practices, then introduce you to something called cure, then introduce you to an example of a cure that I've used, talk about how it relates to discovery and creative activity, as well as student satisfaction, and then remind us that students that come to us at the university have a K to 16 continuum, right? So they've been conditioned previously. And so are there some tools that maybe we could share with our primary and secondary school teachers for tools in the classroom, so that when they come to us, we're all better prepared for meeting this burnout challenge and a student disengagement. Okay, so I'm going to start with a question of, how do people learn, right? When you think about how people learn, or how do they become engaged in something that they don't know anything about, but, they might be interested. right? They might watch something. So I start with the question of how do I learn to play tennis, or how do I learn an instrument? Really, two things maybe that somebody's asking you to learn and you've never thought about it, but it could be interesting, right? Playing the piano, playing the violin, playing tennis. Not something that everybody does, but it is something that people, might like to do. So how would you get started? You might watch something, or you might watch, a person playing, for example, that might inspire you. You might listen to somebody talk about it and say, oh, that's really interesting. I want to do that. You might read about it in a textbook, right? And that could do it, right? You might read an interesting story and say, oh, that sounds like a great experience. But more often than not, the way you become excited about something new is you engage in it. You play. So you might play tennis. Somebody hands you a racket and a ball and you don't know what you're doing. You'll learn the rules later, but you try it out and you really like it. Or piano, same thing. You play on the keyboard and you just explore, you experiment. This is, in my opinion, and the literature tells us that students become engaged more when they're doing it actively. Like that is a way to catch them and grab them. So why can't we think about our classrooms in the same way? Our students are coming in, they really don't know anything about what we're talking about. They may never have thought about it before. And that's the challenge, is to get them engaged, to get them thinking, to nurture them, to do more, right? So how can we use this sort of very simple example of playing, asking them to play, to do the hands-on piece and roll that into what we do in the classroom, right? So some examples of college pedagogy that does this is, something called case-based learning, where students are applying what they know, they work in teams, they work to find solutions, and they report out. And I want to highlight two words here, right? Intrinsic and extrinsic motivation. We're going to come back to that because the literature tells us that if you can tap into a student's intrinsic and extrinsic motivation, that you will be able to hook them, right? Those are two factors. So we'll come back to that later in this presentation. Another example is this thing, right? This pedagogical strategy called project or problem-based learning, right? Where you root the learning around a topic, a very specific topic, right? A project, for example, or a challenge, say on the campus, right, to design more parking and have the students come up with ideas, right? So this is a problem or a challenge, and the students work it through. And another one is self-directed learning. And we hear a lot about self-directed learning in our earlier years in our primary school. In fact, elementary schools do this really well, right? They create play spaces for kids, right? And yet somehow we forget that students can guide their learning, right? So so self-directed learning gives students the opportunity to guide their learning. All right. And in this particular paradigm, the role of the faculty member is to be cooperative, right? It's to motivate students. It's to create access. It's to be available and flexible, right? And to serve more of a facilitator role than a traditional teacher, right? So I just pose this question, right? When we are with our students on the college level, what are we thinking about? When we're teaching in the class, what are we thinking about? Are we thinking about how students are going to benefit from what we're saying? Or are we really thinking about what we want to say? Like what's important to us, right? And both of those are valid and they're valuable, but this is the challenge, right? And of course, how can we do both? Because wouldn't that be great? And this is what we're trying to figure out today, right? So I want to introduce you to something called CURE. And CURE is a college pedagogy that's funded through NSF. I mean, this is sort of their flagship tool. And in course-based undergraduate research experiences also are valuable because they are rooted in faculty scholarship. They really are a way that faculty in smaller schools, schools that are not research one intensive, right? Faculty that have large teaching loads. It's a way that faculty can use their research in the classroom, and students participate in the research as well. And it helps support and promote publications and creative activity. All right. So I want to just give you, two cartoons. Typically, our classrooms might look like this, where the instructor is teaching and people are listening, very much like what we're doing now, right? So have you thought about doing a little less of this and maybe having your classrooms look more like this, where students are working collaboratively, where students are having conversations and discussions, where there's a lot more interaction in the classroom. So students are doing, and you're walking around as opposed to just lecturing to them. All right, so I'm going to give you an example of cure, and I don't know, this could be, this might very well be common here in the, a strategy that's common here in the Bahamas, and I don't know, but I will tell you that it works really great in small schools. It's worked for me. So I want to start, because one of the things about cures is it takes a little longer for faculty to get their work up and going. So in 2001, I was at Yale University, and I was hired to teach a field class. And, I'm a herpetologist. I love snakes, but snakes have very low fidelity. You could be out in the field for hours and days and not see a single snake. However, sea turtles are highly predictable. You know where to go, you know where to find them. And so they make a wonderful case study for students, right? Looking at sea turtle biology to maybe think about big topics like diversity, vertebrate zoology, ecology. Okay, so in 2001, we went out, and this is just an image of me reaching out on the backs of the seat of the Olive Ridley sea turtle, surrounded by colleagues. And you may see the picture here, and maybe you can't tell what they are, but you actually have, right, these little Podocerus nudged in the crevices of the sea turtle's back, right? And so what is that about, right? That becomes the next question. And these are epibionts, right? They are opportunistic organisms that are just found free living on these environment, right? On the backs of the sea turtles in this particular case. Some are obligate, so they attach in an obligate way, and others are inobligate, and I will non-obligate, and I'll talk about that in a moment. I want to share this image not because I'm proud of it, but this is what happens when a sea turtle, a juvenile sea turtle, becomes immobilized and epibionts just settle on the back of it. I want to show you the next picture because this is your juvenile turtles. This is what they look like here in the Bahamas. They're pretty clean. So, that in itself is a fascinating question. Like, where are all of the invertebrates, right? The marine invertebrates, these epibionts, and why are they not attaching to sea turtles, even though we know, right, that sea turtle epibionts are ubiquitous, right? There's lots of them, and they turn up on all sea turtles. We find them, and we also find them on some other marine vertebrates. But today we're talking about the sea turtles. I bring it to your attention not so much to talk about the research and what we've learned, but here is a phenomenon, right? Epibionts, or this host interspecies relationship, right? There's this interspecies relationships of the host and the epibionts. And how do you use this as a teaching tool, right? So case-based, problem-based, self-directed learning. How do you use it? And I'm sure you all have examples in your mind of how, as instructors, you have your systems that you can use for monitoring. Well, this is what I did, right? I can use my system to teach students about biodiversity, to help them define issues that need to be studied, and think about how are they going to investigate their questions. Those are all examples of active learning. I can facilitate that, right? They have fascinating lifestyles. So we start thinking about deeper biological questions in terms of life cycles of invertebrates, right? What's going on? Why is it that you find all stages on the back of a turtle, whereas with other barnacles, you don't, right? And that, comes back to, well, are they sort of living out their life cycle on the back of a turtle, or do you have some free swimming larvae? Again, looking at biodiversity, looking at just the gazillion questions that come out of your students as inquiry. I mean, it's exciting for you as a faculty member, right? To hear things that maybe you hadn't thought about, right? Also, this work made me and my colleagues and my students realize that sea turtle conservation wasn't just going to be about eggs, that there were a lot of emerging diseases that were coming up, and that some of this was being carried by some of these epibionts. And of course, looking at attachment systems, right? This obligate attachment. What did it mean? What were these settlement properties like? So are epibionts opportunistic? Do they just randomly settle on the backs of sea turtles, right? And then you see in the cracks of the barnacle, you find another, right? So we have these secondary settlers as well. So what's going on in these systems? And I'm not here to answer these questions today in my talk. That'll be another talk. You can invite me back. But just to highlight the wealth of curiosity that comes out of a system such as this, right? So now, how does this play into the classroom, right? So in this particular case, this is a research class. This is not individual research. This is where students learn how to do research, right? How do you think of a question? How do you design a question? So how did I use this material monitoring the sea turtles and the epibionts that are on them to guide a course? Because it can't go everywhere, right? You've got to somehow reel it in and let the students lead, but they need to be guided, right? So we had these, large number of samples that had come back of epibionts that were from individual turtles. So what kinds of things did the students engage in, right? Something called morpho sorting, right? Just sorting out what's in the containers. Each student had their own, right? So they could work individually. And then they learned how to use microscopes. They learned external morphology. So they learned skills, they learned characters, they learned details, all for a common goal. They took pictures, they shared their results. You can see if you have a large number of students in your classroom, you can get a lot of data just from your students. Yes, there needs to be quality control, but you can do that, right? You can sort of pay attention to what they're doing, and you're teaching them all the steps. You got to teach them how to use a microscope anyway in the classroom. Why shouldn't they be working on something that contributes to your research as well? All right. So in this particular case, right, what did we do? We were able to get through 106 turtles. That's a lot of turtles, but you can do it with your students if they're properly trained. And the question that the students came up with was, this pre-oil spill, post-oil spill, and I won't get into the details, but this had to do with the Exxon Valdez oil spill that happened in the Gulf of Mexico, America, whatever you want to call it, was in the Gulf. And we were able to draw the line between pre and post oil spill, and students had the opportunity to sort of investigate that. That's what they chose to think about. So what kinds of analysis do they do, right? Because remember, this is an undergraduate course. They're learning about evaluating biodiversity. That was the whole point. And, they really didn't find that they found a lot of things. They came up with a lot of questions. They had a lot of results, but they weren't really sure where to take it next. And it was okay. We ran out of the class. It stopped. They learned a lot of tools. We got a lot of information. Another example of this cure, because remember, it's course-based undergraduate research. My research was on sea turtle epibionts. Now, in another example, invertebrate, like not invertebrate, but invertebrate zoology, which is 204, we introduced another lab that looked at diet of juvenile sea turtles that were in Long Island Sound. And what we were able to do over time, look to see how their diet was changing, how juvenile sea turtle diet was changing. Why? Because we were interested to see how things are changing in Long Island Sound. So we were using this as a proxy to understand how that system is changing. So two different examples, and both are giving us information that will lead to publications, right? And this is where the discovery and the creative activity come up, because you can then have an independent study student, right? In our case, 490, which is individual research, maybe a graduate student, right? Where they can work independently, take the work that was done previously, pull it together, really think about it, add to it. Molly here had also received an REU to go down to Florida, collect another year of sampling, right? So we readdressed this question about the Gulf oil spill and really look at community changes before and after. And that did result in a paper. So it took us some time, but here's an example where the faculty members benefit, the partners who supplied the material benefit, the students were engaged in an actual research project, and a student became a co-author. And just think about the power of mentorship in this entire process, right? So that takes care of three of our responsibilities as faculty: teaching, right? Mentorship and scholarship research. Working with graduate students, right? I don't know, I'm not sure. I would imagine UB has a graduate program, sort of, okay? But if it ever did, this is another way to support graduate education. It also highlighted the importance of regular sampling. It highlighted the importance and the value of, in the classroom, having a system that has regular monitoring, because the communities of change. It also highlighted this work that we did with epibionts, that there was more to be found. So all of a sudden we were looking at invertebrates. And someone said to me at a meeting, hey, have you ever thought about the diatoms? I'm like, what diet? Oh, yeah, I bet there's diatoms. And off we went. We started looking, and we discovered new species. And that led to going to museum collections, surveying the shells of turtles that were in museum collections, and being able to get a temporal understanding of the changes of the diatom community. I thought it was amazing that you could actually get diatoms from turtles that were collected, 20, 30 years ago. It was pretty fascinating. So all of these started from a simple going in the field, working with students, and gradually increased to, right, scholarship, publishing, creative activity, new discoveries, okay? All right, so this is what I said. We're able to bring together all three out of our four main responsibilities. So what is it about CURE, right? It's authentic. There's issue definition, so students get the chance to think about, well, what are the important questions to ask, which is, I would say 75% of our challenge, right, is to get students thinking about what's exciting, what needs to be learned, how do we grow from what we know, how do we take the knowledge and build new knowledge, right? And isn't that what the university is about? Okay. And how great, I mean, they hire us because we're experts. So how great is it when we can actually bring our expertise in the classroom and have it also support our scholarship, right? So this process adds to the existing work of the faculty member. How about identifying student potential? You know, all the students are not the same. So this strategy of a more of an active learning classroom allows you to engage maybe the students that are not as competitive or as knowledgeable, but it also allows you to highlight and identify the ones who are and help them stay excited and move on to other things, right? So it helps you identify student research potential, right? Research is not for everyone, but it really is pretty good for the ones who like to ask questions and do puzzles, right? You're able to scale it to the diverse needs of the students, the learning needs. And I think I talked about that. And of course, at the end, it's publishable. So these are all best practices that have been identified for STEM engagement as well. So not only are we meeting the needs of the faculty, we're meeting the needs of the student. So do the students like this, right? And I'm not going to get into too much of that because that would also be another presentation. But I think it's important for me to share with you, how do I get at that question? This is currently now, the strategy is being funded for me through NSF to sort of use this survey, which I have, and I'm happy to share it with you, that looks at, you know, the different categories, 5 different categories and question students. And you need the multiple questions in the category so you can validate the category. Because a student, if a student is scoring honestly, they should be able to, there should be a high coefficient of scoring, right? They should, if you know, so the first one is, you know, persistence and motivation. Well, if they were motivated, they should be able to score from 1 to 5 in the same way for all of the different categories. So anyway, I will start with 1. You know, you look at the responses from the students and you see that, yeah, you know, did the course include challenging concepts in biology? And the feedback is pretty high. Yes. So most of them agree or strongly agree or agree. So it means that learning in this fashion is significant. The student does feel challenged. All right. That's important because you have people, you know, that don't believe in this pedagogical strategy that'll say, oh, you know, there's nothing really going on in the classroom. They're just playing, right? Well, I don't know. That's not really what's happened. That's not what this is saying here, right? They did feel it was challenging. I mean, you're asking them to read and think quite a bit, which can be uncomfortable for students sometimes. This is the important one. This course improved my confidence to engage in science, and that was pretty high because students, sometimes majors or non-majors specifically, don't like to do science or math, but this strategy engages them, and engagement builds confidence. And I want to just, and I don't have a slide for this, but I just thought of it. You know, cognitive dissonance is so, you know, you've got to get over it. You've got to get a student who's right there. If they love something and they're like, oh my God, I'm such a terrible piano player, but I really love it. And they will get over that cognitive dissonance. They will stay active in it because they want to do it. And you as a mentor are motivating them to stay in it. You're helping them keep that interest. So I love teaching this way because that to me is so important. It's great for, you know, students who do really well, and it's also really important for students who struggle. Well, they come to us from somewhere. They come to college from somewhere. They come in middle school, high school, what's going on there? So I've done a little bit of work on this with middle schools and high schools and teachers. And this isn't really about sea turtle work, but it is about engaging. It is about place-based and project-based learning and bringing that into the high school and the middle schools, making things personally relevant. And I want to introduce you to something that NOAA, which is the National Oceanographic and Atmospheric Administration in the United States, who has this sort of signature thing called MeWEs, right? And MeWEs are these meaningful watershed educational experience. It's another word for hands-on learning, but more specifically, right, students and teachers, right, are picking topics that are important to the school, to the students, to the community, and this is their basis for teaching this aquatic science class, which was our case, right? It was flipping the aquatic science class at the high school, so that it wasn't just a lot of teaching and lecturing, it actually had challenges for the students. And in this particular case, they're thinking about stormwater runoff, which is a problem in a lot of urban areas. I'm sure it can be a problem here too. If the water has no place to go, there's a lot of damage that happens both to stores and to homes. And so it's a challenge. So the students are thinking about this on their own high school. And they're thinking about green infrastructure, rain gardens, right? And that leads to pollinators. And so we bring it in, we brought it into the classroom and empowered the students to come up with solutions for improving stormwater. And they learn a lot. Like, they learned that they are actually living in a watershed, which was a huge, shift in their thinking. I'm going to come back to that. Again, local context is important. It's all part of issue definition. If they can learn to do this in the middle school and in the high school, think about the things that are important. Of course, teachers have to allow them, give them the space to think about these things. When they come to you at the university, right, they're thinking about, how am I going to develop those interests of mine through a university study? So just some results from our high school program in terms of impact, right? 1,200 Danbury High School students, teachers, graduate students, supporting professionals, letting them know that this way of classroom learning, right, with local relevance, hands-on instruction works, contributed to some management plans, contributed to restoration buffers, aquatic plant management ideas, right? Because the problem is fertilizers, nitrates get into the freshwater system. We have algal blooms, which create swimming problems. Anyhow, how do we get at measuring motivation, right? Because that's one of our markers, that this method of teaching, I mean, it all sounds great, but does it work? Actually increases motivation. And there is this science motivation questionnaire. There's 30 questions. Again, I told you that, there has to be subsets because you want to be able to validate responses. It looks at intrinsic motivation, extrinsic motivation, personal relevance, self-determination to learn, self-efficacy, and of course, anxiety. And these are 6 subscales that are thought to define student motivation, right? And so we want to see, can we move the needle in one or all of these categories? And of course, we use a Likert scale to do this. And the important thing is understanding the science gives me a sense of accomplishment. That is one of the questions that then students will read and need to score, never, rarely, sometimes, usually, always. And of course, then we work through these. So what did we find? I think this is what's important. When we got the project started, what we were saying is, aquatic science at Danbury High School is a large non-major science class in the high school. Everyone is required to take science for their high school diploma, but not everybody wants to be a scientist. Not everyone wants to study science, but they have to take a science class. And so a lot of those students will take that. It includes a lot of English language learners. And then we also have the AP, the Advanced Placement Environmental Science class. And here's what's interesting. Remember, they are self-identified. They're going into this AP environmental science class because they and their teachers feel that they can do the work, right? So the question was, if you were to, can you bring aquatic science students up? Can you get them to think like an AP environmental science student? And our results showed, right, at the end of all of this, that the pre-test, so this is when, the first survey, so students come in, right, they're self-identified, they're excited, those scores were lower, right, than the aquatic science post-test. So those students in aquatic science were able to bring themselves, their motivation, their interest, to the level of where the AP environmental students were when they started. If you ask me, I think that's a great success, right? To show that, okay, these students can do it with maybe a different tool, okay? And so, this is just highlighting some of the scores there that also showed us, because along with this experience in the classroom, we also offered a summer experience where we would hire some of the aquatic science students to stay involved with some of these projects over the summer. And that regular engagement with faculty, graduate students, undergraduates on the college campus blew the scores out of the park in terms of motivation. So it really reaffirms everything we know, the importance of, small learning groups, regular mentorship, right? Relevant issues that you're studying, all increase motivation. And, will they become scientists? I don't know, but at least we know that they are interested in some really important issues in where they live. Well, what about middle school? And this is what was interesting. We used the same SMQ. We did a very similar study with middle school students, but of course they're middle school students. So a lot of the work that was done was in the classroom with the teachers. The teachers were trained. And we found that the kinds of activities, like if you really wanted to increase motivation in middle school students, that personal relevance, activities that were personally relevant or increased self-efficacy, right? That they felt a sense of accomplishment for, were the ways to get them to increase their motivation. And I think that was also an important finding. So we know that is one way to act in the middle school, make the information personally relevant, make them feel successful, move them into the high school, give them some real authentic learning experiences. And who knows, maybe on the college level, we'll see them a little more engaged. And of course, as faculty, we have to give them that space, right? We have to let go sometimes of our primarily lecturing style. So in summary, faculty can blend their teaching, research, and mentorship to increase student engagement in STEM, but also increase their scholarship, right? How important is that? Cures, integrate project, problem-based learning, case-based learning, this MeWe, self-directed learning. All of these are active learning examples, and they can focus on faculty research and a faculty member's area of expertise. Natural history collections are invaluable for issue definition and engaging students, and also engaging the public. If you can engage the parents, you're going to engage the students, their kids, okay? Active learning strategies increase all levels of student engagement in STEM. So I want to, with this slide, just acknowledge, right, everyone that I've worked with in the past, because you can't just do this on your own, right? You have to have the partnerships, the external sponsors, the students, everyone, and the museums that are willing to take this material and open their doors to you and your students, right? The teachers, the high schools, the administrators, you need all of that, or this isn't going to work. It's almost like a learning community, right? That's scaled. And I want to end with this and say thank you. And this is a picture of me working on some Bahamian snakes. And maybe the next time I'm here, I can talk to you a little more about my road collision project that I look forward to continuing. So thank you very much for having me, and I'm happy to take some questions.
Any questions here? All right. Check the chats. We can take questions from online as well. Anybody have any questions for Good. Oh,
Yes, hello.
yes, go ahead, Dr. Carmichael.
Right. What I want to ask is the idea of project-based hands-on learning is excellent for those subjects, biology, marine biology, and so on, that have an obvious practical component. Let's go to the bugbear in the room. Mathematics, nice and dry.
Right.
Dr. Pinou, how would you, for example, implement a project-based hands-on learning for an area of mathematics where, for example, you have to explain the issue of division by zero and why that produces the result of infinity? Purely theory, very dry, because what I'm saying here
Tough
is this.
question there, James.
Yes, sorry. So yeah, what I'm going to say here is, when all is said and done, most courses are syllabus based. So there is some set of, there's some set of issues or themes, topics, which have to be exhausted. Project-based learning tends not to always cover every area of the syllabus. But to keep it brief, but to keep it very, very brief, Dr. Pinou, how do
Yes,
you
I'm here.
implement pure or project-based stuff for very theoretical subjects like maths? Thank you.
Yeah, I love that question. Because this is not something that hasn't come up in this, in the States as well. I mean, I don't know, math seems to get a bad rap, right? Everyone's afraid of math. I just published a paper, it'll be coming out soon, on how we can combine science and math together to really promote better math aptitude, right? And so, you and I can talk and I can share that paper with you. The whole premise of that was showing examples of how mathematicians and scientists can work together. Listen, there is no question, if you maintain your class, and I'm not in the math, professional societies. I don't know what the conversations are that are going on with the mathematicians, right? But I do know that there's been some conversation about integrating mathematics more, or integrating the projects and the problems in mathematics, like word problems, to help students contextualize process. And I think part of the challenge is redesigning courses in math as well that are a little more interdisciplinary, so that the students can discover the mathematics. And I know you're probably thinking, oh, well, she doesn't know what she's talking about. And I agree, maybe I don't. but I think can math courses be redesigned so that, what can you do if you know that your students struggle with dividing, by zero? Is there something you could do? Is there like a demonstration or something you could do that, or they do, to sort of discover it? I hope that answers your question. I know you're not going to become a believer over.
No. That was I appreciate that. As a matter of fact, you touched on perhaps the biggest issue, which is, well, this is my personal belief, that mathematics or that math syllabi have or should be redesigned to be more practical, well, to be more project based. But of course, that is a polemic in itself that we won't touch right now. As such.
Right, right. I do believe that, that it's close. I mean, I think that people are realizing this has to happen. What that's going to look like, how do you do it? I don't know, but you know what? I think when there's a will, there's a way, and the math scholars are going to be able to figure it out and work on some exciting things. I think you're going to have to do it with computer science and AI and this whole revolution. Like if we're serious about increasing aptitude in those areas, students are going to have to get over, math fear, in a big way.
Yes, yes, ma'am. Very much so. Thank you.
Thank you.
I think we have just another comment from Dr. Moxie. The same challenge is evident to a lesser extent with other abstract concepts in chemistry, such as MO theory, entropy, etc. Good question, though.
Yes, thank you. And we could talk about that, at another time too. But yeah, I mean, it's just about, listen, I think I think the message here today is all faculty, if you're at a university, you have a research interest. You must, because that's how you got hired. You did something. I mean, you have, PhDs, you worked on projects, you have some passion. And sometimes we're frustrated because our passion gets stalled because we can't motivate and move our students. I mean, we can't be in a vacuum, or we're required to do it, right? We need to publish, or we need to motivate our students. So how are we going to do it? There must be a way to take your research passions in chemistry and mathematics, and I mean, there's professional journals, people publish in this era all the time, right? So there must be ways to sort of integrate students and have them become part of a cure. Yeah, how? Well, I leave that to the disciplinary experts, but I'm happy to, I mean, this is why I'm here. I'm happy to be a sounding board and help facilitate any professor in any discipline who's interested in maybe thinking about how they could do it. I'm happy to help.
Excellent. Any other questions? No? Looks like that's it. All right. Well, thank you so much, Dr. Pinou. That was excellent talk. And we look forward to having some further chats with you.
Well, thank you very much for inviting me. I really enjoyed being here, my visit so far. Thank you very much.
Matthias, did you have? Sorry, Dr. Johansson.
He's just.
Oh, you're clapping. He's sorry.
Just
Out of the hand raised.
by the presentation.
Thank you. Thank you very much. And thanks to my students for attending. I see some of you are here. Good job, guys. You get points.
All right. Thanks, guys. Have a great day. A good week.